Yesterday when I was recording cell division lecture, I was having a lot of trouble with the Loom. I think it was because a lot of people suddenly start using Loom for their online teaching, so the system might be just simply overwhelmed. So hopefully everything is going smoothly. Last time we stopped at crossing over, so I'm going to move on to the next slide. So between prophase and metaphase, you're going to have a transition stage called the prometaphase. Pro means like prior to, right? So this happens right before our metaphase. And if you remember from the previous video, because everything happens in meiosis of 1, so we're going to assign number 1 to all these different stages. And then during meiosis 2, everything will happen number 2. So the key event that happens in prometaphase of 1 is the attachment of spinal fibers to kinetochore proteins. So where are the kinetochore proteins? So you can read this, but in this figure, if you remember, this is after DNA has duplicated, right? So for each chromosome, you're going to have 2 identical copies, and we call them sister chromatids. So over here, where everything really kind of narrows, it's almost like you're pinching the 2 sister chromatids. This spot is called a centromere. So the kinetochore proteins are going to be at centromere. So what happens is you're going to have spinal fiber microtubules coming here and attach themselves to kinetochore proteins, so that during meiosis 1, the 2 homologous chromosomes will be separated, right? So the spinal fibers are going to pull this homologous chromosome to one direction, and then the other homologous chromosome will be pulled to the opposite direction, and they will end up in 2 different cells, 2 different daughter cells, all right? Now, there's a term here. At the end of prometaphase 1, each tetrad is attached to microtubules from both poles. So what is a tetrad? Now, if you look at this pair of homologous chromosomes, how many sister chromatids do you see? 4, right? So because each chromosome has 2 sister chromatids. So together, 1, 2, 3, 4. So that's 4. Now, tetrad means 4. So basically, it means a pair of homologous chromosomes, right? Because it contains 4 sister chromatids. Okay, now this one, just kind of know where it is. It's really not any critical information. So chiasma is the plural, and the singular is a chiasma. So you can just think of it as where the sister chromatids from different chromosomes kind of come into contact. Okay, so this is prometaphase, and then in metaphase, the homologous pairs will be arranged in the center of the cell. Remember, we call that equator or metaphase plate. And then they're going to face opposite poles because they will move away from each other, and they're going to end up in opposite directions, opposite poles. So in metaphase 1, homologous chromosomes will orient themselves randomly at the equator. This is very important. So there might be a question on the test asking you, so when do homologous chromosomes orient themselves randomly at the metaphase plate or at the equator? So the answer is going to be metaphase. And in that process, you're going to have a phenomenon called a random assortment. So what is random assortment? Before I show you that, remember this. This is very important. Random assortment, just like crossing over, can introduce genetic diversity into the offspring. And this is how it works. I'm going to show you this picture. You can use the previous one too, but just for simplicity, let's use this one. Actually, I have a title here, Families Similar and Yet Different. You and your siblings are not exactly the same, right? Even though you come from the same parents. And I'm going to explain why. So let's go back to random assortment. So you have 2 cells over here, and this is a metaphase 1. So you can see that the homologous pairs have a kind of line up at the equator, right? So this is the equator. Now, when they move and line up around the equator, their movement is very random. Okay? So you may have this cell where, let's say, the black represents the father. So the 2 chromosomes, different types of chromosomes, remember? So this, we can call it this chromosome 1, this chromosome 2. So you can see in this scenario, the 2 chromosomes from the father line up on one side, and then the 2 chromosomes from the mother line up on the other side. So at the end of meiosis 1, you're going to have 2 new daughter cells, which will contain different chromosomes, right? So this cell contains the 2 chromosomes from the father, the cell contains the 2 from the mother. And once they undergo meiosis II, the sister chromatase will be separated. These are the 2 gametes that you are going to end up with. So you can see they're different. All right. Now this is only one scenario. What's the second scenario? Because everything is kind of moved randomly, right? So it's very possible that you may have the second scenario where you have the big chromosome from the father, and then you have the small chromosome from the mother. They just happen to end up on one side of the equator. And on the other side of the equator, you have the big red from the mother and the small black from the father. If you have this scenario, you're going to have these 2 cells, right? And then once they finish meiosis II, you're going to have these 2 types of gametes. And you can clearly see that these gametes are all different from each other, right? They have different combinations of chromosomes, so their genetic makeup will be different. So in this case, you can see that if you have 2 homologous pairs, you are going to end up with 4 unique gametes. 4 unique gametes. So the equation is actually pretty easy. I can tell you right now. So that's 2 to the second, that's 4. So this is a fixed number. It's constant. And this represents the number of homologous pairs. Okay? So number of pairs. So if the cell has 2 pairs of homologous chromosomes, if the cell goes through meiosis, it's going to generate 4 different gametes. So what if the cell has 3 pairs of homologous chromosomes? So that means you're going to multiply 3 2s together. So 2 by 2 by 2. So that's going to be 8. Right? What if the cell has 4 pairs of homologous chromosomes? 2 by 2 by 2 by 2, and that's going to be 16. All right. So, this phenomena of different kind of pair up of or different combinations of homologous chromosomes. So this is called a random assortment, and that's going to be the second source for introducing genetic diversity. All right. Now here's my question. We know that humans have how many pairs of homologous chromosomes? 23, right? So you have 23 pairs of homologous chromosomes, and that's a total of 46 chromosomes. So if you are making gametes, so if you're going to make either sperms or eggs, so how many unique gametes can you produce? You know? So we're going to use this equation, all right? So 2 to the 23rd, and it's going to be more than 8 million different possible gametes. Okay? So that means every single one of us can make possibly over 8 million different gametes. They all carry different combinations of chromosomes, so different genetic makeup. So when each gamete becomes a zygote and eventually a baby, that baby will have a unique genetic makeup. Then if the baby comes from another of your gametes, that's different. Okay? Now, what's the chance? Let me go back a little bit. Okay, so let's go back to this question. So families similar yet different. So you and your siblings are 99.9999999% different, right? So you haven't really had any siblings who are identical to you, right? Unless, now there's an exception, unless you guys are identical twins, then you will be different, you will be the same. But for people who are not identical twins, you rarely see any 2 siblings are exactly the same, right? And this is why. Because for each person, we can generate over 8 million different possible gametes. Now, for a couple, this is from the father, right? So the father, the symbol for males, a father can make over 8 million different gametes. And this is for the symbol for for females. So the mother can also make over 8 million unique gametes. So when you take a 1 from the father, that will become the sperm. That's going to be 1 in 8 million, right? If you take an egg from the mother, again, the chance is 1 in 8 million. So when you multiply the two numbers together, the chance that you and your sibling will come from the same gametes with exactly the same genetic makeup is a 1 in. This is a huge number, right? So let's see, this is 1,000, million, trillion, billion, right? So the chance is a 1 in 64 billion. Now I think that's pretty small, right? It's even smaller than hitting a jackpot. But wait, there is more. Okay, so that's not the end of the story. There is another event that will make the number even smaller. So you and your sibling and chances are you are never going to be genetically identical. Okay, remember we still have crossing over, right? That can also enable even greater variety. So if you have exchange of genetic material here between the two chromosomes, but they're not sister chromatids because they're not from the same chromosome, but they're kind of line up right next to each other. So that's gonna further make the chromosome after meiosis I different. So that's gonna further decrease that chance that two siblings are gonna be exactly the same. Okay, so it's a very, very, very small chance. Okay, so I have a practice question. So this question will really test your ability or your understanding, really, how meiosis will work. And especially how the random assortment work. Okay, so here's my question. How many possibilities? So really, I mean, how many unique gametes? How many unique gametes? This is a typo. Would you have if you have 3 pairs of homologous chromosomes? So that means it's 6 chromosomes in total. Can you draw all the possibilities? So I recommend that you just pause here, give yourself a few minutes to draw on your own, right? If you don't do it on yourself, you just look at my answer, you're never going to understand. Because when you just look at something, you think you understand, but chances are that if you actually do it, you don't know how to do it. So pause the video right here. Actually, I'm going to pause it, and then I will draw my answer so that you can look at it later. Okay, so here is my drawing. Hopefully you have done that yourself. Now, so we have 3 pairs of homologous chromosomes. So you can see that we should have 4 scenarios, right? So 4 possible cells before meiosis. So this is the first scenario where you have all the chromosomes from the mother line up on one side, and then all the chromosomes from the father line up in the other side. If you divide that one, at the end of meiosis I, this is what you're gonna have, right? These 3 end up in one cell, and then the 3 blue one end up in the other cell. And then if you go through meiosis II, further divide the sister chromatids, this is gonna be cell number 1, cell number 2, and then divide these 2, and you can see, I'm just going to say this is a possibility 1 because these 2 cells are identical, right? So that's the first possibility, and this is the second possibility where everybody only has the father's chromosomes. One thing I forgot to mention is I don't really have any fancy tool to indicate the 3 different types of chromosomes, so I'm just using the size. So this is going to be the largest one. I'm going to call it big chromosome, and this is medium, and then this is small. And then this is the second scenario. So you can see that when they line up around the equator, the red big, blue medium, and red small line up in one side of the equator. And then you have the other group line up on the other side. So if they divide, this is what we're going to have, right? The big red, blue medium, and the small red. So these are the 2 daughter cells at the end of meiosis I, and then if they go through meiosis II, the 2 sister chromatids are going to further divide. So this is what you end up with. Okay? So this is possibility number 3 because we're looking at the possibilities of gametes, right? And then this is number 4. So can you tell 1, 2, 3, 4 are totally different, right? They have the different combinations of chromosomes. So I only did the first 2 scenarios. So maybe you guys can do the third scenario and the first scenario, the fourth scenario. And then for each scenario, you should have 2 unique gametes generated at the end. So how many total of unique gametes? 1, 2, 3, 4, and then you're gonna have 5 and 6 from this scenario, and 7 and 8 from the last scenario. So the number is correct, right? 2 to the third, that's 8 possibilities. Okay? So if you know how to draw this, then you should have a pretty good understanding of random assignment. All right, let's move on. Anaphase, now things are much easier, right? So the microtubules, the spindle fibers, are gonna pull homologous chromosomes apart. Oh, let me change the color. So this is what's different than mitosis, because in mitosis, anaphase, it's the sister chromatids are pulled apart, right? But meiosis are different because you're gonna do divisions twice. So the first division, what you're separating is the homologous chromosomes. This is a very, very important. There's a good chance that you may get a question on this. So the question may say, in which stage of meiosis do homologous chromosomes are separate or they're pulled apart? And the answer is going to be Anaphase 2. Okay, so sorry, the answer is going to be Anaphase 1. Anaphase 1. I want to show you this comparison between Anaphase 1 and Anaphase 2. So in Anaphase 1, these spots where they're connected, where they're held together, that's the chiasmata, right? If you remember from a previous slide. So in Anaphase 1, you are separating the chiasmata and pull the 2 homologous chromosomes apart, and they will be distributed to do different cells, to the 2 new daughter cells. And the Anaphase 2, which is very similar to mitosis, this time you are going to pull the 2 sister chromatids apart, and this is going to end up in one cell, and this is going to end up in the other cell. Okay, so that's the differences between Anaphase 1 and 2. So let me repeat. If I ask you, in which stage of meiosis do homologous chromosomes are pulled apart, the answer will be Anaphase 1. If I say, in which stage do sister chromatids get pulled apart, get separate, the answer will be Anaphase 2. Right? Okay, so let's keep moving on. So telophase cytokinesis, this is pretty straightforward. I'll let you guys read it and get familiar with it. Right? Now we are entering meiosis 2. Now the main goal for meiosis 2 is to separate, you can guess, sister chromatids. Sister chromatids. Yeah. So what's the main goal for meiosis 1? You should know by now. I just mentioned that. You want to separate what? Right, homologous chromosomes. So I'm going to say big H, homologous chromosomes. All right. So meiosis 2 is very, very similar to mitosis. Pretty much the same. So at the beginning of meiosis 2, each dividing cell has only one set of homologous chromosomes. But remember, for each chromosome, you have 2 sister chromatids. Right? So you are just going to separate those 2 sister chromatids. Again, this is pretty straightforward since it's similar to mitosis. So you can just read this and let me know if you have any questions. Okay. Meiosis 2, there we go, it's about the same. So sexual reproduction. Nearly all eukaryotes undergo sexual reproduction. And why is that? Because it's a very successful strategy to generate favorable traits so that the organism can survive and reproduce better in the ever-changing environment. Okay, so we mentioned this a little bit at the beginning of the lecture. So again, you really need to understand that genetic diversity is good for the organisms, because by chance you may have a very good combinations of genetic makeup that will give you features that will make you kind of at a very advantageous situation than everybody else, so that you can live longer, mate more, and generate more offspring. That's how we define success for living organisms. All right, Now, meiosis and fertilization alternate in sexual life cycles. So we have three main categories of life cycles. So diploid dominant. So the life cycle is dominated by a diploid stage. And the second one, kind of guess, is dominant by haploid. Or for certain organisms, it's an alternation between diploid and haploid. So again, this part is very easy to understand. So again, I'll just leave it to you to kind of read your textbook and get familiar with it. Okay? I'm going to move on to the more important and difficult parts to understand. So genetic variation in meiosis, this is just kind of a repeat of the previous materials. So make sure you read this and understand everything. There will be questions on this. So, for example, I may have a question asking you, what are the sources of genetic variation in meiosis? And your answer will be crossing over and random assortment of homologous pairs during which part? Meiosis 1 or Meiosis 2? Meiosis 1, right before Metaphase 1. Okay, so hopefully this will help you understand random assortment too. I'm just really concerned that a lot of students wouldn't have a good grasp of the concept, so I put more slides and figures over here. Okay. So hopefully you find one of the figures helpful. So the last few slides talk about chromosomal structural changes. So cytologists. So who are cytologists? What do they study? Cytology, cyto means cell, and logy means study, right? So these people will study the cell. So they have categorized numerous structural rearrangements in chromosomes, but chromosomes inversions and translocations are the most common. So you probably are not familiar with this, but the chromosomes actually change and move all the time. So you may have these two types of rearrangements, inversions and translocations. So both have been identified during meiosis by the adaptive pairing of rearranged chromosomes with their former homologs to maintain appropriate gene alignment. So don't worry about this part. I'm just going to show you some pictures of chromosome inversions and translocations. Just make sure you understand what each one is about. Okay. So when we talk about inversions, these are two types of inversions. So inversions, unlike translocations, involve detachment. So certain segments are going to detach from the main chromosome, and they may flip. And they're going to be reinserted into the chromosome. So I'll show you how they work, in a second. So we have pericentric inversions and we have paracentric inversions. They're very different. So pericentric inversion, as you can see in this graph, if we assign letters to different segments of chromosomes, okay, A, B, C, D, E, F, and this blank part, this represents centromere. Centromere, remember, this is the part like where everything kind of gets narrowed down a little bit. So if you have segments that are detached and flipped and go back to the chromosome, that involves the centromere. That's going to be pericentric inversion, right? So C and D, these two sections include the centromere, so they flip. And now when you look at the chromosome, you have a D and a C, so that's called a pericentric inversion. Now, if you have segments that are that do not include the centromere, you have those segments flip, then that's going to be paracentric inversion. So let's say you have here E and F, they flip, so now you have F, E. That will be paracentric inversion. Or if you have A and B, that will exchange, so you have a B and A here, that will also be paracentric. All right, let's see here. So translocations involve the insertion of a piece of DNA from one chromosome into a non-homologous chromosome. So here, we have 2 chromosomes right here. Now I'm going to ask you, are these homologous chromosomes? The answer is no. Remember, homologous chromosomes are just 2 different versions or copies of the same type of chromosome. So they should have the same shape and size. But you can see these 2 are very, very different, right? One is very long and the other one is very short. So they're definitely not homologous chromosomes. And if they exchange segments of the chromosome, that will be translocation. Okay, so this happens to 2 non-homologous chromosomes, while inversion happens within one chromosome. And this happens between 2 different chromosomes, and they're non-homologous chromosomes. Okay, so you can see this part comes off, and it's gonna be exchanged with this blue section. So this is the end product, and you can see that the lengths of the chromosomes are totally different now, right? So previously the blue one's very long, but because it lost that bigger section in exchange for a smaller section, so now it's shortened. And then the previous orange one gets like a lengthened, right? It's much longer than before. So this is translocation. So what if this happens to homologous chromosomes? If you have 2 homologous chromosomes and they exchange certain sections, what do we call that? Crossing over, right? That's crossing over. So that's why the translocation happens to non-homologous chromosomes. Karyotype, just know the definition. So karyotype is the number and appearance of chromosomes. So to obtain a view of the individual's karyotype, cytologists, people who study cells, photograph the chromosomes. And then they cut and paste and sort all the chromosomes, and they make a chart. And the chart is called a karyogram. Okay. So on the test, if I ask you, what is karyotype or what is karyogram, you should know what it is. So here's an example of a karyogram. So you can see that it contains all the 23 chromosomes, right? Oh, sorry, 23 pairs of chromosomes. So it's a total of 46. Now here's my question. First question, is this a female or a male? Based on the karyogram. Female or male? How can you tell? Go to this sex chromosome. So it's 2 X chromosomes. So that means it's going to be a female. Okay. Now, my next question is, does this person have a normal number of chromosomes? So is the number normal? And yes, right? For each chromosome, you have 2 copies. 2, 2, 2, 2. Everything has 2. Okay? So the number is normal. So what if you have an extra chromosome right here? Is that still normal? No. So that means this person's cells have 1 extra copy of this particular chromosome. And if it's chromosome 21, that person has Down syndrome. Okay? Nondisjunction. So what is nondisjunction? So disjunction means you separate the chromosomes properly. So nondisjunction just means that for whatever reasons that the chromosomes are not separated. So this could apply to homologous pairs or the 2 sister chromatids of the same chromosome. Okay? So nondisjunction may occur in meiosis I or meiosis II. So if it happens in meiosis I, which is shown over here, that means the homologous chromosome pair is not split, is not pulled away properly. So as you can see in this picture, this is the equator, right? Okay. And once the homologous chromosomes are pulled away, right, from the equator, there is a mistake over here. So you can see the 2 homologous chromosomes are still together. They didn't end up in 1 cell. Sorry, they didn't end up in 2 cells, but instead they all come to this 1 cell. So after meiosis II, then you are going to have an extra copy of a chromosome, right? So these 2 sister chromatids are from the same chromosome. So instead of a 1 copy, which is the normal number, like this small chromosome, you only have a 1 copy in the gamete. For this chromosome, you have 2 copies, which is not normal. So I'm just going to draw this unhappy face. All right, so that's why you have n plus 1. So because you have a 1 extra copy. All right, now for the other cell, you have you are 1 chromosome short, right? So instead of 1 chromosome in each cell, you should have a big one. So you should have a big one. So you should have 2. But right now, because of the non-disjunction in meiosis 1, you only have 1 instead of 2. So you're 1 short. So the number is n minus 1. Okay, so this is a non-disjunction in meiosis 1. Now, what will happen if you have non-disjunction in meiosis 2? So this is what happens. If you look at the bottom picture, so this is the equator. The equator meiosis 1 separation is good because the homologous chromosomes are separated and they end up in 2 cells. When they go through meiosis 2, so this chromosome is good because the 2 sister chromatids are pulled away, right? But when you look at the second chromosome, you can see that the 2 sister chromatids are not pulled away. So they end up in 1 cell instead of 2 cells. So for this particular cell, you're going to have an extra copy of chromosome. So you're going to have 1, 2, and 3. So again, you are 1 over, so that's n plus 1. And then over here, because you are short of 1 chromosome, so you are going to have there you go. So you can have 1 short, so it's n minus 1. All right. So usually this may only happen to this daughter cell. So this one is the abnormal one. For the bottom half, if everything is separated correctly, then you still have the correct number of chromosomes. All right. Now, if the father or the mother contribute the normal number of chromosomes or contribute the gametes with the normal number of chromosomes, then everything will be okay. But if they contribute the gametes with abnormal chromosomes, especially if you have extra copy, because those cells are more likely to survive and participate in the fertilization process, then you may have genetic disorders. If this is chromosome 21, then that's going to be Down syndrome. Okay? So if you have a 1 chromosome short, well, you have lots of chromosomes, that's called a monosomy, and if you have extra copy, that's called a trisomy. All right. So what is Down syndrome then? So Down syndrome, you have trisomy because you have a partial or complete copy, extra copy of chromosome 21. So you have extra copy, so that's trisomy. The human zygotes missing any one copy of autosome invariably fail to develop to birth. So that means if you contribute a gamete with a monosomy, like 1 chromosome less, because the chromosomes usually carry genes that perform, critical functions. So if you're missing 1 chromosome, you're missing those critical functions. A lot of times, the embryo wouldn't even develop to birth. Okay? So they're not going to become a baby. But if you have trisomies, because you have an extra copy, right? So you may have overexpressed types of proteins. So sometimes the offspring can survive, from several weeks to many years. So in the case of Down syndrome, people can survive a lot longer. Right? Now, usually the extra copies of chromosomes happen to these particular types of chromosomes, 13, 15, 18, 21, and 22. So I actually found some articles on Down syndrome. So I'll show you real quick. So I bet some people will have this question. So Down syndrome is trisomy, but where exactly does that non-disjunction take place? Does it happen in meiosis 1 or does it happen in meiosis 2? So I found this paper. It's very useful. So they did some research, right? And they found that non-disjunction was paternal that means it's from the sperm. So the actual copy comes from the sperm cell. In 9 cases, and was in maternal in 188 cases. So you can see the mother probably is more likely to cause Down syndrome in the babies. And then among these 188 maternal cases, non-disjunction occurred in meiosis 1 in 128 cases. So that means the homologous chromosomes were not separated. And then in meiosis 2, that's 38 cases. So you can see the majority of cases are actually contributed by non-disjunction in meiosis 2. This is actually very, very interesting. And they calculate the percentage. So meiosis 1 era was responsible for 77% of the cases. And meiosis 2 account for almost 23% of the cases. But all those cases are maternal cases. All right. The number, let's see, might be different for paternal cases, but the sample size is really small, right? It's only 9. But let's look at the data. So for paternal non-disjunction cases, meiosis 1, that's 22%. Meiosis 2, it's almost 78%. So it's like the opposite of maternal non-disjunction. But again, I don't think they have a really big sample size for the paternal cases. All right. There's one slide that's about X chromosome abnormalities. So you may have trisomy and monosomy of the X chromosome, which is interesting. I found this website. So triple X syndrome, that means you have trisomy of X chromosome. You can see trisomy X, or how many chromosomes do you have? 37 in total, because you have an extra X chromosome. So instead of XX, as seen in normal females, you will have XXX, triple X. So this is genetic disorder and affects, actually this number is pretty high than I expected. So 1 in 1,000 females. So that's kind of interesting. So this is just additional information. All right. And then I'm going to skip the next slide so you guys can read through it. It's pretty straightforward. All right, so I'm going to stop here.